Welcome to today's talk. I'm going to talk about Hybridoma Technology today. Hybridoma Technology is used for generation of monoclonal antibodies. Monoclonal antibodies. Monoclonal antibodies, as you know, they are the one which are produced by a single clonal population. B cell. Okay, so these are monoclonal antibodies and they are highly specific to their antigen. Okay, so what are the prerequisites for generating these monoclonal antibodies? Prerequisite: you need to have an access to pure or purified antigen. You would require mice. So one was antigen, mice. So what you're going to do is you're going to inject the mice with the antigen. Okay, you immunize the mice, give 3-4 injections like you use for generation of hyperimmune sera, and then you kill mice, harvest spleen and purify B cells. So these B cells are the activated B cells. Which would be generating your antibody of interest. Okay. Now you have activated B cells. You have to fuse them with tumor cells. Okay, so once you fuse them with tumor cells, you use polyethylene glycol as a fusant. You end up with 3 kind of cell populations, that is B cell hybrid, B cell fusion, and then B cell myeloma, that is the tumor cell, which is called as hybridoma because it is a hybridoma. And the third one will be myeloma. Now you should have something to some way of screening these 3 different population of hybrid the cells populations. So for that we use hat selection medium. So once you produce these fusants, then you use hat selection for the selection of your hybridoma. So what is hat selection? Hat selection is based on chemicals that are present in the medium. Those chemicals are hypoxanthine, aminopterin, and thymidine. So this medium here, hat medium, has these 3 chemicals: hypoxanthine, aminopterin, and thymidine. Why you require these 3 different types of chemicals? So you know that cell can make nucleotides by 2 pathways. One is de novo pathway, another one is salvage pathway. Okay, So de novo pathway is right from the start, new, and salvage is by using different end products that are produced inside the cell. They are utilized to make molecules, nucleotide molecules again. So amnoptrin is a chemical that basically blocks this de novo pathway. Okay, so once the de novo pathway is blocked, then salvage pathway is used. Salvage pathway is encouraged by providing hypoxanthine and thymidine in the medium. So these hypoxanthine and thymidine are precursor of nucleotides. So these are readily available in the medium, and then they can be used through a salvage pathway once the de novo pathway is blocked by use of amnoptrin. Okay, so if you remember, we had three different types of populations. Those are B cell fusions, then B cell myeloma, which is hybridoma, and myeloma. So what is myeloma? Myeloma is a B cell tumor. Okay, so B cell tumor. So I want to talk about this myeloma cell, B cell tumor. This B cell tumor is actually deficient in HGPRT or TK. That is hypoxanthine guanylyl phosphoribosyl transferase and thymidine kinase. So these are mutant. Okay, mutant for either HGPRT or for TK. Salvage pathway, for utilization of salvage pathway, you require these two enzymes, HGPRT or thymidine kinase. So that means that myeloma cells which are deficient in HGPRT or thymidine kinase, either of these myeloma cells, they will not be able to utilize salvage pathway to make nucleotides. So ultimately they will die in the HAT medium. B cell will also die because they have a limited lifespan. So when you grow them in tissue culture flask and then you subculture them, after some time they will die. Myeloma cell fusion, myeloma fusions, they will also die because they will, although they are immortal, but as long as they don't have nutrition, how they are going to survive? So they'll also die because de novo pathway is already blocked in all the cells, right? Because of the presence of an amnoptrin. So de novo pathway is blocked, but only salvage pathway can be used. So salvage pathway will work only in this. Why? Because the hybridoma will have genes which have come from the B cells, and it has genes which have come from the myeloma cells. So B cell will have genes for the antibody production, and they will also have genes for salvage pathway, that is HGPRT and TK, thymidine kinase. So both of these will be functional, hence they will be able to utilize hypoxanthine and thymidine, and so they're able to make nucleotides. So hybridoma will survive. They will be able to grow in HAT medium, and they will be making antibodies, and they will be immortal. So we can select these. So we can select these hybridoma cells and culture them and then screen for antibodies. So antibodies can be screened either by ELISA or by radioimmunoassay. So all you need to is antigen of your choice. That is the antigen that was used for immunization, and run ELISA and look for the specificity and isotype of antibodies through ELISA or radioimmunoassay. So you screen these hybridomas for the production of the monoclonal antibodies, look for the specificity of the monoclonal antibodies against your antigen and also the kind of isotype of these antibodies. And once you're satisfied that your hybridoma is producing the right kind of antibody, right kind of monoclonal antibody, then you can grow them in bulk. Grow the hybridoma in large quantities. So you can grow them in vitro in culture flasks, and you're able to take around 10 to 100 microgram of monoclonal antibody per ml of the fluid, or you can grow them in peritoneal cavity of the mice, and you can collect the ascitic fluid from these mice, and there you can take around 10 milligram per ml of monoclonal antibodies, and then these monoclonal antibodies can be used for different applications, either for the diagnostic purpose or for therapeutic purposes. Thank you.